Good afternoon. My name is Arvind Ramanathan, and I am a computational scientist in the Data Science and Learning Division at Argonne National Lab and the University of Chicago. Team Intelligent Resolution, I'm excited to share some of our insights in understanding how the SARS coronavirus 2 replication transcription complex, or the RTC, works. Before I begin, I would like to take a second to recognize the team that stood behind our work. We represent a total of 12 different institutions, including 8 universities, 2 national labs, and 2 companies across the US and UK, as well as the experimental data that was collected in China. I'm particularly proud of what our team has been able to achieve in this short time. It also brings together 4 different supercomputing sites to look at this molecular machine. While the efforts have been dedicated to understanding the molecular details of the SARS-CoV-2 infection process, it is important to pay attention to the entire virus life cycle. Despite the fact that there are effective vaccines available, millions of people are still getting infected. Thus, by understanding the molecular details of the virus's life cycle, we can design novel therapeutics that can potentially cure COVID-19. What you see in this video is a representation of the virus's life cycle. Once the virus attaches itself to the host cell, it enters the cell and releases its genetic material, which is basically the viral RNA. The viral RNA is translated into the respective polyproteins using the host cell ribosomes, which then self-assemble to form what is called as a replication-transcription complex. The RTC is essentially a macromolecular engine, one that can be thought of as a photocopier that precisely assembles and replicates the viral RNA in large quantities. The RTC is also intelligent in the sense that it maintains a high fidelity in replicating the viral RNA and has its own proofreading mechanism. Thus, the viral photocopier consists of several enzyme parts that are referred to as non-structural proteins, which play an important role in enabling the virus to make copies of itself. Prior to the cryo-electron microscopy revolution, observing such large macromolecular complexes would have been really difficult. Thus, the primary scientific goal of our project is to understand the dynamic nature of the RTC while reconstructing its 3D structure by including all of the data that's available. In fact, for the RTC, it is estimated that about 30% of the collected images were unused for the lack of sufficient ability to fit this data to 3D conformers. This data might consist of hidden states or even functionally relevant information for drug discovery and other important aspects, and as a consequence, we need to move from Cryo-EM data, represent them using volumetric representations, and then use all atom simulations to all of the atomistic details. So now we have Cryo-EM techniques, FFEA simulations or continuum simulations, and all atom MD simulations. We then asked how can we bridge this critical gap in terms of filling the missing details within Cryo-EM data. We posit that modern AI architectures can really help seamless integration of the information across the 2 resolutions of simulations in the loop while keeping the experiments and simulations iteratively refined. To keep the continuum simulation self-consistent, we have built novel hierarchical AI techniques that iteratively parameterize and refine and run the simulations at scale. Each of the components of this workflow have different and unique performance requirements which had to be optimized as part of our implementation. To give you more details, Misha Salim from our team will present some of the results in terms of building this complex workflow.
Coordinating these simulation and AI components in an end-to-end workflow is difficult because the resources at our disposal are geographically distributed and they span various administrative domains. I'll describe how we designed Balsam as a platform to couple simulation and learning in a cross-facility pipeline. At a high level, this workflow is distinguished by AI-enabled agents steering the ensembles and wide-area data pipelines that fully overlap AI with simulation. The workflow is initiated by this agent process. This is user-level Python code that runs anywhere and uses the Balsam SDK to manage the workflow by speaking to a central REST API. In the first interaction, the agent provisions compute nodes by creating batch job requests at Perlmutter and Theta GPU. The Balsam sites are authenticated user agents synchronizing with that central service. Here, the sites fetch the batch jobs we created and submit them to the Slurm and Cobalt resource managers on the respective machines. Eventually, the resource managers allocate the requested nodes, and a pilot job called the Balsam Launcher begins to run. Meanwhile, the agent uses the Jobs API to define and submit the distributed NAMD3 simulation ensemble. Jobs contain a flexible resource specification. In this scenario, we're requesting that each simulation occupy a single NVIDIA A100 GPU. The launchers acquire these simulation jobs and map each instance onto an idle GPU. The simulations write to scratch file systems. A Python wrapper of the NAMD3 application triggers asynchronous reporting functions that process the evolving trajectory and write to storage. To move the data, transfer items are then submitted as another resource in the Balsam API. Here, the site bundles transfer items destined for the Globus service, which moves the data across facilities. Once a threshold number of frames have been generated from the two sites, the agent dispatches a model training task to update the parameters in an autoencoder model. As we'll see later, this is just one of the methods in a hierarchy of AI models that ultimately interpret conformational fluctuations of the RTC across length scales. The machine learning runs on a Theta GPU node and persists the trained model weights to storage. Armed with its first set of weights, the agent now enters an inference and training cycle that starts with the forward pass of the molecular dynamics data through the trained autoencoder. The learned embeddings are fed into an outlier detection algorithm, which ultimately ranks the most significant outliers according to some intrinsic statistical score. This outlier metadata is also written back to the storage system. The agent reads the outlier scores to determine which of the simulation instances contain the most novel conformational states. The agent then uses this information to halt specific trajectories that will be reseeded with the more promising outliers. First, the Balsam API is used to send a stop signal to selected instances within the ensemble. After finishing their current chunk of I/O, the interrupted simulations will restart from the novel outlier conformations. This cycle then continues indefinitely, with the ensemble statistics shifting while the agent learns an evolving latent representation of the data. Overall, we can use Balsam as a generic platform to compose cross-facility simulation and learning workflows. Balsam has scheduling, job launch, and resource management interfaces that easily adapt to run on laptops, university clusters, and other leadership supercomputers like Summit, Theta, and Cori. User space deployment on these systems works out of the box, and you control all these sites through a single API. Late binding of the jobs enables migratable execution on elastic resources, and Balsam is robust to task-level faults with customizable error handling hooks. We achieved nearly ideal scaling of the aggregate molecular dynamics throughput in moving from 24 GPUs on Theta to a combined 176 GPUs on Perlmutter and Theta. This is possible by realizing that simulations don't need to block on asynchronous data movement, and likewise, machine learning is able to proceed in parallel with the simulation. In the experiment described here, the agent reseeded the simulations with outliers 5 times in the span of 90 minutes, and the restart operation occupied a negligible fraction of the otherwise busy runtime. Now I'll hand it back to Arvind to take a look at the continuum mechanics.
Thank you, Misha. Now that we have seen the workflow section in detail, let's now understand how we ran our continuum simulations using fluctuating finite element analysis. The FFEA methodology represents proteins as a viscoelastic continuum solid. As such, the multimeric SARS-CoV-2 RTC is represented as a blob that you can see here in the revolving movie, which is formed of nodes, tetrahedral elements, as well as triangular faces. A simple Lennard-Jones potential proportional to the overlapping volume of the interfaces are used to evaluate the point-wise stress. The continuum motions are evaluated as a function of the density, the velocity, the viscous stress and elastic stress, which capture the material properties of the protein, as well as thermal noise that accounts for the intrinsic fluctuations accessible to any particular region of the blob. This thermal noise parameter is a key distinguishing factor from standard finite element analysis techniques. The interactions can include pre-computed potentials as well as steric interactions. The external forces are the ones that keep the blob together. These are exactly the parameters that we estimate from our all-atom MD simulations as they operate on individual tetrahedrals, keeping the simulations consistent. The restraints on the RNA components as well can be obtained from the all-atom simulations, which will be later described in the talk. There are several innovations that went into the implementation of FFEA here. While FEA, the finite element analysis approaches are very common in modeling airplanes and other large surfaces, these structures are well defined. However, biomolecules are inherently flexible. This means that we need to shrink the macroscopic finite element analysis down to the nanoscale by including such thermal fluctuations. The ultra coarse graining of the continuum level significantly smooths the free energy landscape, relative to atomistic descriptions, and therefore speeds up the exploration of the conformational space by nearly 100 to 1000-fold. Now that we have described the FFEA simulations in detail, John Stone will explain how all-atom simulations were used in the context of our workflow.
Thanks, Arvind. NAMD and VMD are molecular dynamics simulation and analysis tools developed by our laboratory at the University of Illinois. Both programs were heavily used, and they each incorporated innovations important to this project. All-atom molecular dynamics simulations of the RTC included over 1 million atoms for timescales long enough to capture important conformational changes. Simulations in the million atom size range are difficult to scale effectively on conventional distributed memory hardware platforms due to the impacts of internode communication latencies. To address this, we've developed a new NAMD3 GPU-resident simulation mode that exploits GPU-dense hardware platforms with tightly coupled GPU memory interconnects to reach longer timescales for simulations in this size range. VMD was extended to provide the analysis and visualization capabilities needed for FFEA simulations, in addition to its existing features for all-atom molecular dynamics simulations with tools like NAMD. In order to fully benefit from GPU-dense compute nodes found in machines like NERSC Perlmutter and ALCF Theta GPU, we have revised NAMD to bring the same cubic patch-based data and work decomposition strategies from the distributed memory implementation into a new GPU-resident simulation mode that takes advantage of direct peer-to-peer GPU memory load store and atomic memory operations. Interatom forces and updated atom positions are examples of the types of data that are exchanged among the GPUs during each simulation time step. At a high level, we can think of the key communication patterns here as multicast and reduction operations that exchange and sum these data among the GPUs. The new GPU-resident simulation mode shifts the entirety of the core molecular dynamics simulation loop onto a series of GPU kernels. All of the molecular data needed by the force calculation and integration phases of the simulation loop remains in place on the GPUs, thereby eliminating host GPU data transfers. In the new GPU-resident mode, the host CPU threads manage the execution of GPU kernels, handle special NAMD features, and migrate atoms among the GPUs as needed as they move from one patch to another. This slide summarizes the NAMD3 multi-GPU scaling performance for the RTC simulation running on an 8-GPU DGX A100 compute node such as those in ALCF's Theta GPU system. The solid green line with triangles shows the scaling performance for the 1.1 million atom RTC simulation. The blue line with circles shows the scaling of the 2.2 million atom RTC simulation on the same hardware. The black horizontal line indicates the performance for the 1.1 million atom RTC simulation running on 128 nodes of the TAC Frontera supercomputer. The performance of 3 GPUs on the DGX A100 matches the performance of 128 nodes of Frontera for this size problem. Here we can clearly see the performance benefits arising from the use of the tightly coupled GPU hardware with the new NAMD3 GPU resident scheme. With 4 GPUs, we observe about 70% scaling efficiency for both problem sizes, and this is the GPU count available on NERSC Perlmutter, where our large-scale workflow runs were performed. For larger GPU counts, we can see that scaling begins to taper off due to the PME long-range electrostatics algorithm, which incorporates a 3D FFT. We plan to address this using a task-parallel PME dedicated GPU approach and by using a more scalable multi-level summation electrostatics method. VMD was extended to support analysis and visualization of FFEA models and simulation trajectories, thereby unifying access to all of the different simulation modalities used in this project. We've also incorporated a completely new VMD visualization mode based on real-time ray tracing. The new ray tracing mode replaces OpenGL rasterization and allows scientists to view molecular structures and dynamics with high-fidelity lighting and shading in all of their interactions with the program, making it much easier to produce the kinds of movies and figures shown throughout our presentation. Back to you, Arvind.
Thank you, John. Now that we have seen the heavy hitters of our computational workflow, let me explain how we designed hierarchical AI approaches that explicitly couple the continuum and all-atom simulations. The FFEA simulations, as well as the all-atom MD simulations, define an implicit hierarchy capturing the overall fluctuations in the RTC conformational landscape. To keep the simulations self-consistent, allow them to driving each other in a complementary manner, we devised a hierarchy of AI tools that enable us to describe the global to local and back to global conformational fluctuations within our multi-resolution simulations. The first of these methods uses what is called as anharmonic conformational analysis enabled autoencoders, or ANCA-AE. The ANCA-AE approach effectively acts as a hybrid linear and nonlinear dimensionality reduction tool and can automatically identify the global conformational changes that are captured within our continuum simulations. The details of the approach are described in a recent paper. However, the outputs from the ANCA-AE includes novel conformations from which we can steer all-atom MD ensembles. The variational autoencoder focuses on the individual subunit level dynamics of the complex. This uses a deep clustering methods where we input the protein contact maps and is then used as a variational autoencoder to learn a reduced dimensional space. What we have shown that method works very well on small proteins, scaling it to accommodate individual subunits whose contact mass can actually be about 1K by 1K can still pose significant challenges. Finally, the MD integration steps are quite computationally intensive. And as a consequence, we prototyped a novel surrogate model, what is called as graph neural operator, or GNO, which examines short timescale transitions and can basically predict the outputs of an MD simulation without having to run the costly integration step. As Misha explained in the workflow section, these techniques are integrated within our workflow to enable near real-time feedback to the simulations, such that the two simulation scales can be self-consistent. To examine the individual subunits, we selected the 4 helicase parts from the RTC and used the GNO model to summarize the largest conformational changes as captured by the model. There are 4 helicase subunits, or NSP13 subunits. The latent space learned by the GNO model on the MD simulations of the helicase is visualized using the TSNE method. It consists of both the NSP13 1A and 1B subunits located on opposite ends of the RTC. While the NSP13 1B does not undergo a lot of motions, NSP13 1A does undergo a lot of motions, and hence it is represented using a larger sphere. Each conformation from the all-atom MD simulation is represented and painted by the root mean squared deviation, which is just a measure of the overall conformational change with respect to the first starting structure of our simulation. What is interesting to note is that the GNO-MD clusters similar RMSD structures together, and when visualizing the motions, one can also observe that there are different areas of the helicase that are acting differently across the entire RTC. In the last portion, we present an overview of how we studied the scaling profile for the variational autoencoder on our ALCF AI testbed using the Cerebrus CS2 architecture. The scaling profile for the CVAE model measures the throughput as samples per second against the number of GPUs available. On both Summit and Theta GPU, we observe near real linear scaling. However, there is a drop in the throughput as the contact map sizes increase to about 1K by 1K. Interestingly, the Theta GPU provides about 4 times the throughput on the single GPU itself compared to about the V100 GPUs on Summit. When we do scale the CVAE model to the CS2, which is just a single engine, we observe a speedup of nearly 100x with respect to the single V100 card versus about 28x with 8 A100 GPU cards on the Theta GPU nodes. The performance measures are out of the box and not optimized for the CS2 specifically, which could even boost the throughputs further.
Thanks, Arvind. We will now talk about how we integrated the workflow with the Cryo-EM data and obtained novel biological insights into how the RTC functions. Even for massive complexes such as the RTC, experimental methods give us areas of the protein that are well defined, but as can be seen from these Cryo-EM images, there are other portions of the protein which are not fully resolved due to current limitations in resolution capabilities. Therefore, we need methods to add clarity to these areas, which are sort of hidden in the Cryo-EM data. We start from the best initial guess of the Cryo-EM structure using FFEA simulations, as well as models with all-atom resolution, which give us sufficient resolution to study fluctuations and behavior of the individual subunits of this large complex. We use all-atom simulations to study the unwinding process of the RNA and its interactions with the protein, which is one of the crucial functions of this complex. Here, the RNA has to reach the active site of the NSP1014, responsible for the proofreading of the replicated RNA nascent chain, which is important because unless we get quantitative insights into this process, it will be difficult to design small molecules that will stop the viral replication altogether. When bringing back the entire complex, we now have information about the interactions and forces driving these dynamics, and we can use these and provide feedback for the FFEA simulations by refining the input parameters for these protein-protein interfaces. This will more accurately describe the global motions and dynamics of the complex at much longer timescales. So here is a view of these forces acting on the protein-protein interface coming from all atom simulations, which keep the FFEA trajectories faithful to what we observe in the all atom simulations. Also, due to the longer scale allowed by FFEA, we can observe biophysical phenomena such as dissociation or reassociation of protein subunits. The RNA unwinding process is not easily observable because it occurs in such a restrictive protein environment. Even some of the best experimental structures lack parts of the RNA or they're in the unwound conformation. But more importantly, because experimental methods provide static images, they're not able to capture the process or the dynamics, which is essential for understanding this mechanism. We do, however, have information about the active centers, which act upon the RNA for proofreading, so we can use the advanced features of NAMD for non-equilibrium simulations to model this process to give insight into the path the RNA would take to reach these functionally relevant sites, as well as the conformational changes that are induced along the way in the protein. About 3.5 months of work went into modeling this process by determining the right combination of non-equilibrium methods, and we succeeded by using collective variables to pull on the RNA to the right sites, while restricting the path that the RNA can take by employing the MDFF grid forces and also conserving the double helical structures with extra bonds. This is the first simulation of its kind, modeling the RNA unwinding, and therefore we provide significant insights into this mechanism. This is only doable with the NAMD engine because of their capability to apply different methodologies. So what we learned is that all atom simulations and the FFEA trajectories provide complementary information in terms of the complex dynamics. At all atom level, we observe significant motion in the helicases to accommodate for phenomena such as the RNA unwinding, as well as the slight stabilization after the RNA has reached the active sites, indicated by the lower RMSF values. On the other hand, the FFEA subunits provide us with information about the interaction and rearrangement of the NSP1014 subunit, which is the proofreading complex, and the RDRP, which is responsible for catalyzing the RNA synthesis. There are more dramatic changes in the NSP1014 subunit in terms of motion, whereas the RDRP is not as mobile. When looking at the Cryo-EM structure, the root mean square fluctuations indicate that we don't see a lot of motion because this is the result of aggregating an ensemble of conformations while having discarded 20 to 40% of the data due to poor resolution. But clearly, this data could contribute to the description of these important motions which the complex undergoes. So why is all this work relevant? Well, while much of the efforts are being focused on prevention of infection by targeting the spike protein, there are still millions of people who get infected either because they don't have the opportunity to get vaccinated, but even if they are immunized, they can still get infected. So there's an urgent need to address the post-infection treatment options. Another advantage of looking past the spike is that, as we've witnessed in the past couple of years, the spike has mutated several times, which means continuously having to update our treatments. But for the RTC and specifically the RDRP, mutations are much less likely. There have been efforts to adapt past successful RDRP inhibitors, for example, from SARS-CoV to SARS-CoV-2, but while they have shown great promise in experimental settings, the results were not replicated when employed in clinical trials. So first, we have to find ways for the drug to move inside this large complex, and second, even after overcoming the crowded environment, to target the correct active sites to inhibit function, and therefore, really understanding the intermediate steps of the mechanism can elucidate pathways towards this goal of stopping the virus from replicating itself. We established high watermarks for what can be achieved via our multi-resolution simulations. We were able to build a workflow spanning multiple geographic sites. The NAMD simulations have been ported to GPUs and now provide significant speedup in terms of simulation time. Novel AI methods coupled the two scales of simulations, but most importantly, now we have the ability to reverse the flow of information and refine and complete gaps in the experimental data. We have seamlessly combined a variety of methodologies to be able to provide novel insights into the SARS-CoV-2 RTC by showing the unwinding mechanism of RNA and providing context for a lot of details still unavailable experimentally. This data will serve years of research into drug discovery for the inhibition of the RTC. The simulations alone set a total aggregate of 59 exaflops, with another 4.35 exaflops contributing from our AI methodology. We now have the capability to run this workflow at sites where the experimental data is being generated, such as the argon photon source, and we can thread them to simulations that may be running at multiple other locations to take advantage of the latest and best architecture for specific tasks. We have now demonstrated in preliminary work that the surrogate models that we have designed for MD provide speedups for at least 100 times as shorter timescales. What this brings about are certain requirements that need to be placed in the context of emerging supercomputing architectures. One, the immediate need for remotely interacting with and transferring large volumes of data, both from experiments and simulations. Two, the need for visualization toolkits that enable exploratory data analysis at scale. And three, the coordinated placement of heterogeneous architectures with easy programming models that can scale AI training and inference modules without having to optimize for every architecture. We would really like to thank all the people who have funded this work, the computing time which was generously made available, and our colleagues.